Hello, and welcome to All Things Considered, a podcast where we discuss important issues with fascinating guests through the lens of critical thinking. I'm your host, Christopher DiCarlo. On today's show, we're going to talk to Dr. Charu Chandrasekera. She is the founder and executive director of Canada's first and only centre dedicated exclusively to alternatives to animal testing. This is located at the University of Windsor, and it's called the Canadian Centre for Alternatives to Animal Methods. I heard Dr. Chandrasekera recently being interviewed, and I thought she would be a fantastic guest for our show because as a cancer researcher, she discovered something rather unsettling about the use of animals in testing. And when I first learned about her project and her dedication to finding alternatives to animal testing, the reason why astounded me. The statistics about the use of animals and the transition between the value of that testing towards humans was absolutely astounding to me in terms of what I discovered. So I think you're going to find this to be very interesting indeed, and you're going to find her to be a fascinating guest. I'm going to run through a little bit of a bio here, and every now and again I'm going to stop and I'll ask you to elaborate on it. We can have a lovely conversation about what you're doing.
I'm looking forward to it.
Good stuff. All right. Charu, you received your doctorate in biochemistry, molecular biology from the University of Calgary, specialized training in cardiovascular molecular biochemistry. It says here you utilized a full range of molecular, biochemical, pharmacological, and physiological techniques to lead research projects on dilated cardiomyopathy and cardiac ischemia, reperfusion injury, I hope I'm pronouncing that right. Subsequently, as a research associate in diabetes and endocrinology at Wayne State, you conducted research designed to elucidate cellular signaling pathways regulating glucose homeostasis in mouse models of diabetes. Okay, can you please break that down?
It was essentially conducting biomedical research, trying to understand the molecular basis of human heart failure and diabetes using a number of different techniques, including animal models, mostly mouse and rat models of heart failure and diabetes. One of the laboratories that I worked in, we created heart failure in these mice by essentially tying off their coronary artery, reducing blood flow to the heart, and over a few weeks' time, they will go into full-blown heart failure. Right. And we also had genetically modified animals where we deleted certain genes from their cardiac myocytes, from their heart cells. So they would show various signs of dilated cardiomyopathy, heart failure, and have reduced heart function. So we did a number of different things trying to understand the molecular basis of human heart failure using these mouse models.
For sure. Now, I have a number of close friends of mine. Their father was diabetic. He died of a heart attack. Why? Why is the heart affected when somebody has diabetes?
There are a large number of different factors, but in general, diabetes increases the risk of heart disease by about tenfold. A number of different mechanisms involved, glucose homeostasis, right? Maintaining that regulation, how you control blood sugar is really important. Glucose is the currency in our body. This is how cells communicate with each other. This is how cells get energy. So when that homeostasis is essentially dysregulated, it affects a large number of organs and a heart especially.
So is it related to like pancreatic issues, and then is it a matter of metabolizing?
Well, it's a number of, so in diabetes, especially in type 2 diabetes, your pancreas is affected, your insulin is not regulated properly, you have insulin resistance in your skeletal muscle, your liver glucose homeostasis is altered. So there are a large number of and then there's, communication between different organs, your kidneys, you could get nerve damage from it. There are a large number of physiological responses that are altered as a consequence.
Right. Now, somewhere along the lines in your career, something happened to you in terms of using animal models. I heard an interview with you on Quirks and Quarks on CBC Radio 1 and was fascinated with the way in which you're trying to do a bit of a paradigm shift in modeling for medical research. You're the founder, correct me if I'm wrong, you're the founder and executive director of Canada's first and only center dedicated exclusively to alternatives to animal testing, the Canadian Centre for Alternatives to Animal Methods, or CCAAM, and its subsidiary, the Canadian Centre for the Validation of Alternative Methods. And these are both located at the University of Windsor. Can you tell us a bit about these organizations, how and why they were started, and what you plan on accomplishing?
I mean, the ultimate goal is to change the Canadian animal testing landscape, to really shift away from using animals as the primary mode to understand the molecular basis of human disease and for consumer product safety testing. That's the ultimate goal, is to really end animal testing in the long run. But the way it got started was when I was doing this heart failure and diabetes research using mice and rats mostly, but I was also exposed to other laboratories with heart failure models in dogs. So I saw it all, I did it all. And during this time, I experienced firsthand the futility of animal research. 95% of drugs tested to be safe and effective in animals fail in human clinical trials. And there are a lot of other issues as well, reproducibility crisis, as you may have heard in science, where you can't have the same mouse model in two different laboratories and get reproducible data. There are lots of different issues. And these animals, they don't really capture the human disease accurately. They may give you a snapshot of what's wrong with a human, but it does not accurately capture that complex human biology, because we're obviously different, right? When you're talking about mice versus dog versus humans, we're a species separated by millions of years of evolution. And there's no simple way to really reconcile those biological differences. And so I experienced this firsthand when I was doing this research, but I went into that research thinking I was going to help humanity. I wanted to really help cure heart disease, learn more about this disease, and contribute to drug development, all of that stuff. So what happened was, about 4.5, 5 years into this work, my dad had a heart attack that required quadruple bypass. And right after his surgery, I was sitting at his bedside at the Halifax Heart Center, and I was looking at him, and I was looking at everyone else in this ward, and I was thinking to myself, the work that I'm doing, is that really going to help cure heart disease, help people like my dad and all these other people in this ward? So what I was doing at the time was working with these genetically modified animal models where these mice lacked certain receptors on the heart cell membranes.
Right. Can we talk about that for a sec? Are these called knockout mice?
Yes. They're called knockout mice.
Tell us how they're genetically modified. Like, there are factories, right, that do this? You order these things.
Essentially, there are companies that, there are companies that will do it for you. If you tell them, I want you to create, send me a mouse with X, Y, and Z genes deleted, knocked out. Or in the whole body, or just knock it out in heart cells or pancreatic cells or liver cells. So you could make these different types of knockout mouse models, or you can replace a mouse gene with a human gene. So you can get all these, you can do it in your own lab, or you can get someone else to make it for you. I mean, now it's really quick. You can order it online, and within a few weeks they'll be there for you. So yes, I was working with these knockout mouse models of these receptors that play a critical role in the heart. And it was thought at the time that if they were activated in time during a heart attack, then it will reduce the damage following a heart attack. So this is what I was working on. And the whole time I was sitting beside my dad, I was wondering, were these receptors activated in his heart? Did it minimize the damage? All of these things, right? It's natural to think about this. So a few weeks later, when I came back to my lab, I was talking to this experienced career cardiovascular researcher who's worked on these receptors, who's been in this field for 25 plus years. And I ran into him and I said, do you think these receptors were activated in my dad's heart during his heart attack? And he looked at me with this, I'll never forget this look. I don't know how to describe it, but he looked at me and he said, how the hell would I know? We've never looked at this in the human heart. And at that point, something changed within me. You know, for the last 4.5 years, I'd already experienced the futility of animal models and all the problems with animal research. But at that point, something had this, his comment had a profound impact on me.
Yeah.
And I thought,
sure.
I did not go into heart failure research to cure mice of heart failure.
Yeah.
And I walked away from all of this shortly after.
Now, what strikes me, and I'm sure our listeners and viewers, is hard for me to fathom. Hearing this for the first time, 95% of research on rat and mouse models is non-transferable to humans. Why am I hearing about this now? Is this something commonly known? Is this something that you guys are keeping hidden from the rest of the world? Like, that blew my mind when I heard that figure, as I'm sure other people are wondering, how is that even possible?
Well, it's not surprising, and it's possible when you look at the differences between the biology between humans and animals. And, you know, I mean, this fact has been around for a long time, but we have a scientific culture that's ingrained in animal research, a culture that truly defends animal research. So it's not very common. It's not the most common thing for people to be going around spewing out these failure statistics of failure and other things. We always, as a scientific community, we tend to defend animal research. We glorify the limited successes and we completely disregard the colossal failures. That's probably why it's not, you know, on every newspaper. And then even when you think about scientific publications or even an article on CNN, people often talk about, oh, there's this research from X university that cured this disease. And you have to read halfway down the article to find out that it was done in mice or dogs or monkeys and not humans. So it's the culture that we have in place at the moment.
Wow. And do you have any knowledge of when these models began being used, like historically? I mean, we can look this up and we can fill people in on that, but this has been going on for a long time, correct?
It's been going on for many decades. So the last 100 years, I would say, the last century, we've focused a lot on animals. And some of these animal studies at the very beginning, if you think about the discovery of insulin, for example, right? Banting and Best, they used dogs, but that's what they had available at the time. And they made these history altering medical discoveries. And most of the textbook knowledge that we have currently came from animal studies. So animals have made monumental contributions to the life sciences. Mm-hmm. We've used them for the last 100 years, but over the past few decades is when we really ramped up with these technologies to knock out certain diseases. So with genetic modification, and as technology progressed in our culture, in our society in general, we also found quicker and faster ways to manipulate these animals to create all these different disease models. And then on the other hand, when consumer safety came into question, and when you have, you know, over 80,000 chemicals on the globe today, we needed a way to find out that they would be safe for humans. So over the last few decades, we've seen a huge increase in animal testing.
And the trend doesn't seem to be waning a great deal. So if I'm a scientist and I want to apply for a huge grant, am I going to have to put somewhere in there that I'm going to be using animal models, or are they going to take me less seriously if I try to work around that?
This is a very interesting question. And in reality, you shouldn't be penalized. You shouldn't be questioned if you don't want to use animal models, but you are. Every major funding agency, the National Institutes of Health, Canadian Institutes of Health Research, and other such federal funding agencies in different countries, and scientific journals, they almost always ask you, where is the animal data? And God forbid if you had data from humans that happen to contradict what you've already seen in animals. They question the human data, not the animal data. That is the state of affairs in modern day biomedical research. So it is very difficult. I personally have received comments saying you need to validate your 3D bioprinted human tissue model using a mouse model. And this is very common among, colleagues in my field, that we're constantly fighting against this. No, I don't want to give you animal models. For one thing, we have a lot of animal data that we've accrued over the last few decades that we could technically compare against them. We don't need to do new animal studies for many of these things. But it's a question that always comes up. It's almost like science is incomplete without animals.
Right. This is interesting because, this whole show is based on critically thinking about important issues. You're bucking the system. You're questioning the dogma of established science. So a lot of people look to science as being this collection of people who all agree, who all kind of think the same way, and there's peer review, and they really don't see, to borrow a metaphor, they don't see how the sausage is made. They don't understand the nuances behind getting funding, what takes place behind the scenes with different scientists who might disagree. With this new concept of altering models and going to alternatives outside of rat and mouse models, what kind of pushback are you getting? How are your colleagues finding this? Do they think way to go, this is wonderful, or you're crazy, this is career suicide, you shouldn't be doing this? What has been the reaction within your own community of scientists?
It's a set of mixed reactions. Obviously, there's a group of scientists from different sectors, so from industry, from academia, from government, right, like Health Canada, FDA, European Chemicals Agency. There are researchers and scientists in every sector that fully support alternatives to animal testing. There's been a lot of change in this field, especially in toxicology, where you're testing pesticides and cosmetics and household cleaners on animals. There's a huge shift in this area with changes in the way we do the science. There's new innovation, new ethical standards, and legislation requiring you to move away from animal testing. And there are national strategic roadmaps to move away from animal toxicity testing.
Right.
However, there's another group of people, and especially when it comes to studying human disease, what we call biomedical research or hypothesis-driven research, where animal models are still the absolute gold standard. And you're obviously, there's a lot of pushback. Oh, I can't do it. I cannot do this without a mouse. I cannot do it this way. I cannot do it that way. You need this. You can't see full organ interactions. You cannot get this full physiological basis of life without a living, breathing animal. You cannot do it in a petri dish. I mean, there's so much about why you cannot do it. And a lot of people are saying, well, until I can replace it all, I'm not going to change anything at all.
Interesting.
So there's a lot of different, and you're required, as I said, when you're trying to apply for a grant, you're trying to publish your data. Where's the animal data?
Exactly.
You need to validate your human data in an animal model. All of these things. So there's a mix. There are those who are willing to accept it and move forward in trying to advance this field. And then the ones who are still stuck in, this is the way I've always done it mentality, and resisting change.
So in this case, it's not great to be old school.
When you think about it, right? Like the other areas of human our society, how long did it take from going in horse carts to landing on the moon? It didn't take very long, just a few decades if you think about it. Why aren't we implementing that kind of change in this field? It's kind of mind-boggling, truly, why there's so much resistance, because science is an evidence-based enterprise. It's not a belief system. The evidence is clearly showing you that animal models are inadequate. They're antiquated. They're not the best way to capture human biology. Yes, we've learned from them, but we need to now move beyond them if we truly want to make a difference for humans who have been waiting for decades, right? Alzheimer's, Parkinson's, ALS, muscular dystrophy, cystic fibrosis, cancers, heart failure, diabetes. We do not have cures for any of these diseases, but we've cured them all in mice.
Now, I might be able to help. That sounds audacious. But here's how I see things. So in critical thinking, I get students to draw out what their arguments actually look like. There's a tool for diagramming what a person's argument looks like. And at the very top, it's going to look like a house where the roof is the conclusion and the walls are the premises in support of that. So if we had both your argument and their argument side by side, and your conclusion is you don't need rat and mouse models, and their conclusion is, yeah, you do need them. Then if we showed the reasons for your argument next to their reasons, could that visual, do you think, could that extend maybe a bridge or a capacity for them to visually see the difference between the two arguments? Might that be a helpful tool in some regard?
Of course, I mean, we could do that again, but that exercise has been done many times over, right? The evidence has been compiled in many fields of science. So if you were to take even a particular disease field like Alzheimer's disease, for example, it's been over 100 years since Alzheimer's was discovered. We spent enormous amount of time and energy studying this disease, and over the last decade, there's a 99.6% clinical trial failure rate. We don't have any disease-modifying drugs for Alzheimer's disease. But what does the scientific community want to do right now? They want to create a better animal model. So all those arguments about why these animal models do not work exist, and not just for one disease, but in general. But we have a culture that tends to overlook and sort of undermine them, hide them under the rug kind of attitude. So no matter how strong the arguments are, some people just don't want to accept them. But it might, be very well worthwhile to put it into that sort of structure as you describe it.
Okay, well, that leads right into the next tool in the critical thinking skill set, which is biases. Why are people unable to get past their biases? And while we're talking about that, maybe we should think about how to strategize to account for or accommodate people's biases so that they have the capacity to maybe better understand why they're having difficulty accepting new information that challenges established dogma. Do you have any hypotheses as to why some people, tell me it's not money, right? Just tell me it's just they're not doing it simply because it gets them more money or notoriety.
No, money.
Because that's not a scientist.
No,
then.
I don't know. Money is not the only reason. I mean, you could get money to do alternative models too, right? But I've thought about this a great deal. I don't really know. I just think that it's human psychology, cognitive dissonance. I don't know what to call it. It must be a whole number of reasons. And this is the same thing, not just in biomedical research, but if we look at our society as a whole, why do we hesitate, right, to accept people with different beliefs, or to let go of certain cultural practices that have been in place for millennia? We have, as humans, we have that tendency to sort of lag behind in accepting new things. Right. And I think it's just amplified in some way in biomedical research. There are other industries where we've moved away from certain technologies, right? They've become obsolete. We've moved on. We've accepted the new methods. But I honestly don't know what the reasons are, other than it's something that people are really comfortable with, and they feel at this point, all the replacements are not there. And therefore, I'm just going to wait, or it just cannot be done. But it's not really based on facts.
Right. Philosophers like William Kingdon Clifford and David Hume, these people, they spoke about what's called the ethics of belief, which gives us this notion of what's called epistemic responsibility, which is, have you done your homework, and should you be believing this? Because in so believing this, it will affect the way in which you act. And you could be acting incredibly in harmful ways because you haven't done a sufficient amount of epistemic responsibility in attaining that information. Science is supposed to be the pinnacle of knowledge. And it amazes me that you're telling me the community isn't getting on board as quickly as might be expected. So are they caught in a form of groupthink? You mentioned cognitive dissonance, sweeping stuff under the rug and so on. But is it out of a matter of convenience? We've always done it that way, so we should just kind of keep chugging along that way. Or I'm too old to change, can't teach an old dog new tricks, right? So I'm deeply concerned about the cognitive level and the biases and the context in which all of this is happening. And if I can hear what you're saying and think, wow, if that's true, what do we do? Why do we continue doing this? We need to halt this ASAP, because you're just bringing about harm and discomfort to sentient animals that you don't need to do. So stop it. I'm having a really hard time trying to wrap my mind around this kind of pushback. And if it's not for money, and it's just that's the way science has always proceeded. Well, science is an organic changing what have you done for me lately process, right? It has to follow where the evidence leads. And you understand, as a critically thinking professor, if the evidence is there, we are obligated to follow where the evidence leads. And to not do so, you're literally not being a scientist. So I'm having a really difficult time trying to understand how scientists aren't accepting of the evidence. Is there a disconnect somewhere? Is it just not publicly known within the communities?
So the facts are known, right? Anytime you publish an article, you're doing your research, you are expected to acknowledge the limitations of your study. So oftentimes, even in these animal studies, when you read through papers, whether it's a Nature paper or a Cell paper or a Science paper, a high-profile journal or not so high-profile, whatever the medium may be, you always have to acknowledge the limitations. So the limitations have been known for a long time. People acknowledge them, but there is this systemic change that's been very difficult to bring forward. So if you are a researcher at the University of Toronto, McMaster, wherever, if you are a researcher at a university and you are in this, publish or perish culture, you're going to do what that culture requires you to do. Not many people have the opportunity to fight against that and to win and to be able to do their science. So we don't have a system in place that incentivizes this type of research. If you are applying for a grant from NIH or Canadian Institutes of Health Research and they're asking you to provide animal data, not many researchers want to really fight against that. They want to keep their lab open. They want to do the science that they love doing. So this change really has to come from top down, where funding agencies actually mandate non-animal methods, and then bottom up with researchers like myself who just said, we're not going to do animal testing anymore. We're going to, start embracing these new technologies. So the change has to come from top down, bottom up, and a complete overhaul of the system, which is happening. It's not happening fast enough for my liking, but it's happening, but very slow.
Okay. Now, are you still director with Physicians Committee for Responsible Medicine?
No, I'm not. No, since I started the Canadian Centre for Alternatives, I have not been in that position.
Does the organization still exist?
Yes, absolutely. They do a lot of great work in the US.
Shouldn't everybody be on the Physicians Committee for Responsible Medicine? Like, to me, that just sounds like, yeah, of course. I don't see any Physicians Committees for irresponsible medicine. So I'm just wondering, why does that have to be necessary? Is that not built into the very concept of medical research? Or am I just naive or stupid? I'm just not getting how things actually function in the real world of science.
It's interesting while I'm talking to, someone like yourself, with expertise in critical thinking and philosophy and all of these things. I don't really have any solid answers as to why. It's something I think about constantly, but it's a combination of these factors that there is a certain way that it has been. And changing that is just difficult. That's the thing. If you had certain standards put in place, perhaps legislation requiring you to. This is what happened with cosmetic animal testing, right? In the European Union, as soon as the legislation came into effect that, cosmetic animal testing will be banned by such and such a date, companies, everybody came together and they tried to figure out how are we going to do this, because by 2013 we're not going to be able to do this anymore. And that's the same kind of. I think sometimes humans just need certain limitations and restrictions and
Right.
conditions, and then you find a way to do it. Right?
Interesting. Yep.
If you're
Yep.
in that situation, you're going to figure out a way to swim upstream and get it done.
Yeah.
But we are not in that situation. We have
Right.
academic freedom. We have the freedom to do whatever we want for the most part as a scientist.
Mhm.
And therefore, there's no huge incentive to do that.
I'm wondering about the similarities between what you're trying to do and another historical case. I'm not sure if you ever heard of Dr. Ignaz Semmelweis. He was an Austrian physician in the 1840s, and about 10% of the women in the maternity ward in the main hospital in Vienna were dying of childbed fever. And he didn't know why. And with the midwives, there was only about a 1 to 2% death rate. And this was very embarrassing to the medical community, who were at the time all men. And they had no idea. They thought it might be, miasma, which is like vapors in the air, that kind of thing. Some even said it was the will of God. What can you do? It was their time, and God needed to call them home. But then Semmelweis was doing an autopsy, and he handed his colleague a scalpel, and it cut his finger. And he said, don't worry about it. He kept going on with the autopsy. 3 days later, his colleague dies exhibiting the same symptoms that women had, dying of childbed fever. So he realizes there's something in these dead bodies. And after physiology and anatomy class, we go right to the maternity ward and examine the women. Maybe there's something passing from the cadavers to our hands to the women. And he called it cadaveric matter. So he had all of his physicians wash their hands in chlorinated lime, because he figured whatever this invisible stuff is, this should surely kill it. Of course, the death rate goes way down. He publishes, he goes to the head of the hospital, and he says, we have a whole new science here, germ theory, essentially. And they fired him because, what are you talking about? Germs. We can't see them. He tried to get his material published. It got rejected. He had what was then called a nervous breakdown. He was committed to an insane asylum, and he was kicked to death and died from sepsis, and went largely unrecognized until, of course, Joseph Lister and Louis Pasteur found his books and said, of course, this guy, he's bang on. I'm finding it really interesting that it sometimes takes, and the same story is true with plate tectonics and continental drift. That theory was laughed at for quite some time. When Peter Singer, the philosopher from Princeton, proposed animal ethics in 1973, philosophers laughed him out of conferences, and they're not doing that anymore. How long is it going to take, do you think, for people to come on board and start to, it seems that shifts in science can be glacial in terms of, or like steering the Queen Mary. It takes a long time to get them to happen. But I find it so disheartening, because I teach my students, trust me, just follow where the evidence leads. That's what scientists do. And now I'm having a really hard time hearing that scientists aren't following the evidence where it leads. I don't know. I'm not asking you to fix it overnight, but I'm just wondering, how long do you think this is going to take to shift this paradigm away from these kind of useless models?
So it is happening.
It is? Good.
No,
Good.
we're talking a few decades, I'm going to say, maybe 3-4 decades. I'm really hopeful that I will see the end of animal testing in my lifetime. And the change is happening, and it's a little slow, and it's faster in some fields. So what happened was that in 2007, the U.S. National Academy of Sciences had a committee that came up with a visionary report. It was actually chaired by a Canadian scientist, Dr. Dan Krusky at University of Ottawa. And it was called Toxicity Testing in the 21st Century, a Vision and a Strategy. And this vision was about a future away from descriptive whole animal testing to one that embraced more human biology-based methods. And since 2007, we've seen leaps and bounds of change in that field. So use of animals for chemical toxicity testing. There are countries that have now come up with strategic plans. The United States, for example, they have 3 different strategic plans to shift away from animal toxicity testing. There's legislation that's enacted by the U.S. Environmental Protection Agency that requires them to shift away from vertebrate animal testing for looking at chemical safety. So, and then in the European Union, it's the same thing. Last November, the European Parliament adopted a resolution calling the European Commission to come up with a strategic plan to phase out all animal testing. So these things are happening, but some of this is heavily focused in only one area of animal use, which is toxicity testing. Where we're seeing this big gap and reluctance and resistance is in biomedical research. This is mostly researchers at universities with so-called academic freedom to do as they please. So that area is really lagging behind. But I think looking at that toxicity testing vision, we could apply the same methods, same models, same kind of legislative processes, same kind of innovation, and make a lot of progress in that field. It's happening. It's slow, but I think another 2-3 decades, we should be able to see significant change led by the toxicity testing community.
Right. Okay. At this point, we're going to have to bring in some Latin terms that you scientists use. You're advocating for a paradigm shift in which our species, rather than animal models, become the quintessential model in research. And you promote integrative human-based research frameworks, incorporating every level of human complexity from DNA to the entire organism, that can serve as the primary platform from which hypothesis-driven basic science discoveries are launched. And you emphasize that through widespread use of currently available and newly developed human-based in vitro, ex vivo, in vivo, and in silico methods, scientists can garner greater replication and translation that benefits the understanding, prevention, and treatment of human disease. In vitro, it's Latin for in glass, correct?
Essentially, in a test tube, right? Vitro is glass, but it's essentially in a test tube or a petri dish, so outside of the body. Yes, capturing human biology in a petri dish or a test tube is essentially meant by in vitro. And the other terms are the ex vivo is where you're talking about taking, for example, a human organ out of the body. So let's say you're going through a cardiac transplant, a heart transplant for somebody suffering from heart failure. You can take that diseased heart during that transplant, the heart that's sick, take it back to a research laboratory, make it beat again, and you can infuse that heart with drugs, and you can look at how it beats. You can essentially bring it back to life in a laboratory, and you can learn so much about the genes that are altered in that diseased heart, how it contracts, how all the biochemical signaling that's involved in a beat-to-beat contraction, how certain drugs can react. So those types of things where you can take a human tissue out of the body and do something with it is often known as ex vivo. And they are what we call organotypic tissue slices. You could even take a very small biopsy of an organ or some tissue and do a lot of work with it. You can, honestly take one single cell and learn so much about your DNA, your RNA, your proteins in there. There's a technology called omics technologies, genomics, where you're looking at your entire gene expression profile of hundreds and hundreds of genes, how they go up and down under different disease conditions. So there's a lot of different ex vivo methods that you could use with human tissue samples from surgical remains or from cadavers. And then in vivo is where in vivo inside the organism, where you could do non-invasive imaging, right? Positron emission tomography, MRI, functional MRI. There are lots of different non-invasive methods where you could monitor drug interactions inside the body in real time, and even things like biomonitoring, which exist in pretty much every country. On planet Earth right now, where governments really take samples from people, right? Whether it's a urine sample, blood sample, breast milk sample, and you can look at how your body reacts to environmental exposures. These chemicals that are in your environment, they get into your system, they're processed a certain way, they're eliminated a certain way. So from collecting simple human waste, whether it's a blood sample or urine, all of these nail samples, hair clippings, you could learn so much about how humans react to long-term exposures to chemicals, long-term exposures to multiple, exposure routes, whether you were ingesting something or you're putting something on your skin. So there's a lot of what we call human epidemiological data that you can obtain from certain populations, and you can monitor some of these populations for changes. There are some ethnic groups on this planet who are resistant to certain diseases. What can we learn from their biology, and how do we apply that to develop another drug for somebody who has an increased susceptibility to a certain disease? Right. So in vivo methods are, it's not about testing drugs on prisoners. That's not what it is.
Right.
It's really
Right.
about capturing human biological complexity with consenting human subjects who are willing to give you, certain kinds of information. And the other in silico. Silico comes from essentially computer chips, right? Silicon chips. That's where the term is derived from, computational modeling. I mean, think about it. The artificial intelligence, computational modeling that we have today, they could surpass any animal testing. There are simulations that you would never be able to do in even an animal.
Let's talk about that, because that has me really excited. So just for our viewers, in vitro, in glass, in a test tube; ex vivo, outside of the organism; in vivo, inside of the organism; in silico, computer-based models. I do a bit of storm chasing for Environment Canada. I chase tornadoes when the weather gets severe and whatnot. There are some people working in Wisconsin now, University of Wisconsin. They developed the most sophisticated computer models available in the world, cutting edge stuff. All they did was put in the parameters of a weather system, and you should see the storms that they can develop on their own in real time. It's absolutely mind-blowing. With the type of AI that we have available in conjunction with quantum computation, are we going to see a move in that direction where we don't have to cut open animals anymore? We can put in the parameters, we can put in the constraints, and we can ask the computer to do certain things on its own, given what those instructions are. How far along are we in those models? How accurate are they in helping us?
Oh, they're incredibly helpful, and we're already seeing tremendous progress in that field. So in reality, we have, especially in chemical toxicity testing, there are tens of thousands of chemicals for which we have a ton of animal data already. So if you are a company that's developing a new drug or a new chemical that's going to go into a hand cream, you can look at thousands of chemicals for which we already have data. You can see if your new chemical has a structure that's similar to one of the other ones that already exists, and you can kind of predict from there, if our chemical structure was a certain way with these certain chemical groups, how would it potentially, what category would it belong to, and how would it potentially interact? What kind of adverse effects would it have in the liver? And this is also based on lots of human data that we have acquired over the last few decades, and also existing animal data. So you could make these structure activity relationships, and you can model some of these functions. And artificial intelligence, there's so much data out there. We have tens of thousands of scientific publications showing how certain pathways, biochemical pathways, are activated and inactivated in a cell. So you could use AI technology to sift through these, and there are lots of different databases that government agencies and companies and industry have developed that are even publicly freely available for people to go and extract a lot of information out of, and you can do all of these things. And that field is just burgeoning. You're only going to see an explosion of different models and utility for using that to inform your decisions on your risk assessment or on a disease condition.
I remember hearing physiologists and various medical scientists talk about how difficult it was to conceive protein folding. And now it's done in computer models, and it's blowing their minds. I don't know if you ever saw that image of the first one they ever did the computer simulation of it. It is incredible to see. And I'm very hopeful that we can focus more on these types of in silico types of models in the future. It's far less invasive, far less necessary to use animal models. I read an article in which you were apparently watching participants engaged in like a Canadian Cancer Society run. And you said, I wish I could tell them they're not running for a cure. They're running from a cure. What did you mean by that?
Oftentimes when you're looking at these charities and you're donating money, you don't know where that money is going. It could be going, I mean, obviously some of it is going for patient care, which is really important, right? Community-based support, all of that. But the funding that goes into research, it goes into animal research, just like with funding from National Institutes of Health or Canadian Institutes of Health Research. The vast majority of that money goes to animal research. And I wish some of these people truly knew that the money is going to this research that isn't really making progress for that field. You can continue to work on mouse models of cancer until the cows come home for another century. You're not going to find the cures that have eluded us for the last 5 decades. If you're using the wrong model, and that was the thing. I remember sitting in that cafe in Halifax talking to this journalist, and the CIBC Run for the Cure was happening outside. And I almost wanted to tell them, when you donate money, ask the Canadian Cancer Society to put it towards non-animal methods. Could you, have an option? I often tell people when they ask me that if I'm going to donate to a charity, I want to make sure that my money goes to something that is going to make a real difference, and putting more and more money. If you think about the Ice Bucket Challenge for ALS, it generated tens of millions of dollars. Do we have any effective treatments for ALS right now? We don't. We're nowhere near finding a cure for ALS or even effective treatments that could modify the disease. That's really what I meant by that. It's difficult sometimes to watch because the public is not aware about the extent of animal testing and what goes in research and how ineffective it is. So it's frustrating at times to watch that because people have been misled, or they're just simply not aware, and they think that, if you can experiment on 1 million rats or 1,000 monkeys to save my daughter or my grandmother or my father, then go ahead and do it, because I don't care about those animals as long as you can find a cure. That's sort of the prevailing beliefs that we have in our society.
I know. That was actually my next question. I had a colleague who once said exactly that. I don't care if it takes 1 million rats, to die. As long as it saves one human life, it was worth it. What if people listening or viewing, are thinking along the same lines? Aren't humans top of the food chain? Aren't we the most important species on the planet? Why shouldn't we just kill whatever we need to if it's going to benefit us?
I don't think of us as, at the top of the chain. I think that we're part of this whole planet. We're part of the animal kingdom. And we need to have that common sense, right? We're primates with big brains. We need to use our large brains, use our rational thinking that got us to where we are today, to truly change what we're doing for humanity and for other species on this planet. It's a matter of common sense too. in some ways. And the thing is, we have experimented on tens of millions of animals, and we have very little to show for it. And that's the thing. So are we going to continue to do this, or are we truly going to go through a rough period, embrace change, and start focusing our energy somewhere else? And this is where, sorry, I've been fiddling with this for the last little while I was talking to you. This is this new technology. So this would be considered an in vitro technology. This is organ on a chip. And this is essentially a chip the size of a thumb drive. You can see that I'm holding it here. And you can plate small amounts of cells in these little compartments that we have and essentially capture a microcosm of human physiology in this thing. And this is now being used, is being, the US FDA is testing this technology for drug safety testing. And this is going to revolutionize the way we do chemical testing and drug testing over the next decade. And the work that we do in my laboratory too, we're creating 3D bioprinted human tissue models, and this is essentially what they look like. This is sort of a dried up tissue that's on this petri dish, but we got these petri dishes with these different compartments, and we're developing kidney model, GI model, heart, lung, liver models, where you could capture human biology in this little thing with a couple of million cells.
Huh. Interesting. Now, the Netherlands are going to end animal testing for chemical safety by 2025. Where is the rest of the world? Where are they at this point in time? Are they leaders? And who's lagging? And where's Canada in all of that?
The 2025 deadline has since changed.
Okay.
That came out back in 2016 or 2017 when they made those goals, but that has since changed slightly by another decade or so, I'm going to say. And then back in 2019, the US Environmental Protection Agency said the same thing, that by 2035, they want to end toxicity testing on animals. But those deadlines are not going to be hard and fast. They're going to change a little bit. But in general, yes, the European Union and the United States, they're on a very good trajectory to phasing out chemical testing on animals. And in Canada, we're making some progress. I'm going to say that we don't have the kind of legislation or financial investment in this field, or even, an Animal Welfare Act that is protective for the animals in science in Canada. We don't have any of that, but we are making smaller changes. The Canadian Environmental Protection Act, for example, CEPA, that was amended recently in the Senate, contains language requiring you to reduce, replace, and refine the use of animals for toxicity testing. And once that goes through the House, hopefully we will have that finalized in legislation that we need to move away. And I think the Liberal government in their last election platform did mention 2035, just like the US EPA did, that it would be a good time to phase out chemical toxicity testing. But we don't have that kind of, it's not hard and fast written legislation yet. So we need to make similar changes for studying human disease as well. Chemical testing is a whole other ballgame, but understanding, instead of making knockout mouse models of Alzheimer's disease, when are we really going to focus on using human Alzheimer's disease in a dish, or heart failure in a dish, that type of thing.
So when you approached the vice president of research at Windsor back in 2016 with a proposal for the Canadian Centre for Alternatives to Animal Methods, the centre now works still in three main areas: biomedical research, regulatory testing, and developing courses and degrees focused on training the next generation to think outside the cage. So how are your students responding? What has the reaction been? Are some students like, no, we need animal models. That's the way we've always learned. We have to keep going this way. Or are they accepting? Are they accepting of it?
Fortunately, the next generation is far more open-minded than the current generation or the previous generations of scientists. So I'm very hopeful. No, they're very excited. I mean, look students today, they have an iPhone 13 in their pocket. Why would they want to resort to animal testing?
Right.
You think about like the very first computer it took up a whole room, and now you essentially have your whole world at your fingertips in a device that's so small. It's the same kind of change. They understand that these technologies like organ on a chip and 3D bioprinting, they are excited. They can see the possibilities, and honestly, it's inevitable. It's going to be their future. So most of my students, every time I give a lecture somewhere, or the ones that I'm training here at the University of Windsor, they're excited. They want to learn more. And even if they cannot completely get away from a certain animal studies, they want to try and incorporate some of these new methods. And it requires a different way of thinking. So if your primary way of thinking is, I'm going to take this mouse, knock out a gene, and see what happens, it's not the same kind of reasoning and rationale that goes into taking 3D bioprinted tissue models to mimic a certain disease. It requires a different kind of mindset and experimental design. And these students are doing it very well. One of the exercises I did last year with my class, so we currently offer Canada's first graduate course in this field. It's called the Human Subject, Animal-Free Methods in Biomedical Research and Toxicology. I gave them one assignment and I asked them, if you had to rediscover insulin today in 2022, how would you do that without using a dog model? And some of the answers were amazing. These are master's and PhD students, and the kind of experiments they designed and developed, it was just so refreshing to see that they were open, that given the chance, that they wouldn't resist.
What about pushback from some who say that in vitro models for some diseases, come a long way, but the majority of animals used in biomedical research are a necessary part of preclinical toxicity or carcinogenicity studies, since humans are eventually going to be receiving these treatments eventually? Wouldn't leaving out those intermediary steps of animals end up endangering or killing human subjects?
This is, it's an interesting argument. The thing is that there's a 95% failure rate. It's already happening. It's not like we're keeping everybody really safe by testing on animals. And the argument is that a lot of people are waiting for this magical technology that's going to replace it all, that's applicable to each scenario, but that's not how it is done. And my answer to many of these researchers who are willing to consider anything beyond animal testing is that what we have at our disposal is a versatile toolbox that has a large number of tools that you can configure anywhere you like, depending on the question that you're trying to answer. So it's not a matter of just, if you can't do it all, that you're not going to do anything at all. But what we have is a toolkit, perhaps none perfect as standalone, none that fits all. But if you use the whole toolkit, instead of just the hammer, you could study human biology in a different perspective. And that's the only way to really reconcile this. It's not really simple to go from knocking out a gene in an animal to capturing Alzheimer's in a petri dish, but it's being done. And when you think about especially the progress that we made in toxicology, so one of the big examples, and this is a procedure that's now approved at the Organization for Economic Cooperation and Development. So the OECD has a chemical test guidelines program. And the 35 OECD nations, including Canada, participate in this test guidelines program. And last year, they approved this one procedure that completely replaces mouse and guinea pig testing. So for something called skin sensitization. This is where you get a skin rash or an adverse reaction, an allergic reaction in response to repeated contact, skin contact of a chemical. This is normally, and this has been for the last few decades, been done with mice and guinea pigs. Those tests were completely replaced by essentially 2 human cell-based tests. This is how simple it can be sometimes, and it required a completely different mindset. It required a completely different way of thinking to address that issue. How do you figure out if that chemical is going to cause an allergic reaction. And people did it by working together. Different disciplines came together, and now it's a simple, what's known as a defined approach, simple approach, where you take a chemical, you put it through your first test battery. Actually, the very first step in that procedure is in silico. It's a computational model, and you have your chemical, you put it through the first test. If it's positive, you do a second one. If it's negative, you can essentially be confident that it's not going to cause an allergic reaction. This is how sophisticated it can be, even with a simple cell-based assay. You just have to be open-minded and be very creative to develop what we need to capture that human biological complexity. It's doable. There's no excuse for it.
Are there some things in medical research for which we just simply can't get away from animal models? Like in the development of the COVID-19 vaccine, were animals used, and were they necessary, or we couldn't have made it as quickly? Or are there alternatives in that form, in that type of research as well?
So if you're talking about replacing all animal tests like tomorrow, no, we're not going to be able to replace all animal testing. There are some, biological phenomena that we haven't quite been able to reproduce yet. But that's not to say that in 10 years' time or 20 years' time that we won't be able to. And yes, while we have some of those things that we are still trying to develop, we also have a lot of other things that, even if it's not one single method, as I said, that could replace what you want. Maybe there are 5 different methods that in combination will give you enough information about the biology underlying what you're looking at. So yes, we are not ready to end animal testing tomorrow morning in certain areas. And one of a specific example that I can think of is some of these in vitro methods, right? These tissue models, for example, are lacking blood vessels and nerve innervation. So the nervous system controls a lot of physiological responses. So how do you incorporate that? And so the next generation of these models, that's exactly what they're doing. Even in our 3D models in my laboratory, we're incorporating blood cells, and trying to create that interaction. And then organ-organ interactions. They're with these chips, they're combining multiple chips together now to create human on a chip. So you will have your kidney, liver, GI, heart, brain all together connected.
So with in vitro, will you be able to somehow replicate and manipulate the metabolic and hormonal activity and cell structure that can basically reliably recapitulate what is seen in humans? Do you think we're close? We're there?
That's happening already, and people are incorporating immune cells into these systems. Some of these companies that have sophisticated organ chip models, they've already incorporated immune cells, so you can see immune cell recruitment into these tissues, depending on chemical exposure or disease condition. Blood vessels being incorporated, nerve innervation, hormonal signals. Yes, it's all happening. There's a lot of progress that's being made. I mean, the next 10 years, I think, it's really exciting to see what's unfolding right now.
It is. It is. Tell us about your cat.
What can I tell her? My cat changed my life. She transformed my life in a way that no human has ever done. So at the time when I was going through this difficult time as a scientist, witnessing the futility of animal research, at home, I was going through a personal transformation with this magnificent cat who walked into my life. Her name was Mowgli. And with her, I'd had other animals before, but Mowgli had a profound impact on me. She taught me firsthand what animal sentience is all about, that animals are just like us. They feel pain, joy, they're mischievous, they get mad, they have the same set of human emotions. And I witnessed this through her. And every time I looked into her eyes, there was this innocence and purity in her eyes that captivated my heart. I saw a person there. She was not the automata, the machines that, Descartes mentioned in the 17th century. This was a being. This was somebody with a biography. This was somebody with life experience who made choices. So with her, I became very, very attached to her. And that bond that I had with her, I started witnessing that in the laboratory animals. So one day I remember going to the laboratory, and I was just about to euthanize this animal to take a mouse to take his heart out for experiments. I brought the animal up from the animal vivarium, put it in the lab, and I was, just grabbing the solution to euthanize the animal. I turned around and I looked at this animal, and it was just like cleaning its mouth, kind of like what cats do, right? Just like licking the paw and then trying to clean its hide. And I stopped and I looked, and we made eye contact. And at that moment, I saw the exact same expression of innocence and purity in that animal that I saw in Mowgli. And I think, I would forever be grateful to Mowgli for changing my life that way. She's the reason she was a big part of this. Just realizing the failures of animal research wouldn't have been enough for me to walk away from it all. So Mowgli played a profound role in my life to realize, to think about the value of ethics, upholding the highest ethical standards. And that's something that truly changed my life.
I had a similar experience. Kind of an interesting story. My wife's not thrilled about mice and rats. She has this kind of phobia. And so my son Matt and I went off to the pet store to buy a gerbil because she doesn't like rats, but gerbils she could kind of handle. And at the pet store, the person said, "You're not going to get along very well with a gerbil, but rats are very social." And so she talked us into getting the rat. My wife wasn't thrilled, but learned to adjust. And, they don't live very long, a couple of years. But when I had to take it to be put down, because it was clearly at the end of its life, I cried more for the death of that rat than for either of my parents when they died. And the reason was because I talked to my parents long before they died and asked them, "Have you had a good life? If you died tomorrow, have you had a good life?" And they said, "Yes, of course." And so they lived a long life. They lived a full life. But having a rat look you in the eyes when you're taking to kill it, essentially, and that level of innocence you talked about, that's extremely moving because you're in full control of this animal's fate. It can't escape. It's not right. It has no choice in the matter. And it led me to wonder, to what extent are we generally bringing about pain and discomfort to sentient animals unnecessarily? And so that's why when I first heard about you and what you were doing, I thought this is great that somebody's doing this, because when I was teaching at the University of Guelph, a professor in psychology, Hank Davis, every year would have a service, a secular service, in honor of all of the lab animals that gave their lives for humans. And I thought that was bang on. Like, that was a really impressive, evolved way of understanding the relationship between us and other animals. And now I have a dog, and it's even more elevated because even greater capacity to show signs of emotion, properties of discomfort, comfort, love, disturbance, and whatnot. And we do learn an awful lot from these animals outside of what we can get from them in the lab. What do you say to people who look at people like us and say, you're just a bunch of hippies. You're just a woke crowd that changed the world in your kind of hippie views? Or what would your message to the world be?
My message is that we, as Homo sapiens, we have a tremendous amount of responsibility in our hands to be the stewards on this planet, to take care of all these species, not just laboratory animal species, but all inhabitants on this planet. And we should be doing better year upon year, decade upon decade, century upon century, to uphold the highest ethical standards. The ethics must be a part of every aspect of our society. And with especially within science, the animals cannot and must not be treated simply as a tool in science. They're living, breathing, sentient beings who, if they had the choice, would never consent to experimentation. And we have a huge moral responsibility in our hands to make sure that we minimize animal testing. Yes, we may not be ready to replace it all tomorrow morning, but we have an enormous amount of responsibility as an intelligent species capable of critical thinking and reasoning to do everything in our power to move away from animal testing. There is no other way to advance our species and advance life for everyone and for anyone without doing this. We're not just doing this because it's an animal rights thing to do. We're doing this for ourselves as well. And I have one question. I'm going to read something to you on my phone. There was this tweet that I randomly came across back in April. And this is not a woman that I know. But it just, when I opened up Twitter for my center, I saw this tweet. She says, dear followers and other people, this is my last tweet. In a few days, I will receive the final injection to end my life. Multiple sclerosis as one. As a scientist, as a human being, what would you tell her? That we've cured it in mice, but we don't have anything for you, that you have to end your life this way? If you don't have an answer to that question, then you should not be doing animal research. That's how I feel about this, because it is unfair to not just the animals that we needlessly create this miserable life for, but also the humans who have been sitting around for decades waiting for a cure that's thought to be around the corner. So we have a huge moral responsibility for our species and for the ones we are meant to protect on this planet.
Well, hopefully, moving forward, we'll see greater focus and attention towards that end. There will be a genuine ethics of belief and greater critical thinking used in how it is we go about doing research moving forward. Listen, I want to thank you very much for joining me today, and I wish you every success in your future and endeavors and what you plan on accomplishing. And I hope to be able to help in some way. So thank you very much.
Thank you so much for having me here. It's such a pleasure to speak with somebody from a different field who has a fresh look on this, as an outsider in some ways. So I've learned a lot from you as well. And I do hope that we can continue to be in touch and work together to sort of get this to be the norm, for human biology to be the gold standard.
Absolutely. Thanks again.